Thank you very much. It's an honor to be with you today, especially following such distinguished speakers. I would like to present to you today the work that we have been doing at QCRC on the genetics of cardiomyopathies with a focus on dilated cardiomyopathy. So the topics we will go through today, starting with a definition of DCM, discuss with you the rationale of our strategy, the methodology we have been using, importantly, the results today, and where do we go from here. So dilated cardiomyopathy is a disorder of the myocardium. Patients present with increased dilatation and decreased systolic function. You can see a representative diagram here. There are millions of patients affected worldwide, and that costs billions to the healthcare systems. Importantly, over 50% of those cases have a genetic basis, and this is what triggers all the genetic studies that are important to take place. So why is it important to decipher the genetics of dilated cardiomyopathy, and why pursue this program at QCRC? Well, because understanding the cause of the disease, so what's going on at the DNA level of those patients, of those individuals, we will not only identify more accurate diagnostic markers, and importantly, pre-symptomatic diagnostic markers that will give us power to predict which family members are at risk to develop dilated cardiomyopathy in the future, but it will also give us information so as to make more accurate prognosis, because different mutations may lead to different outcomes, and last but not least, to develop better targeted treatments, because if you know what the cause is, you can target, correct, and lead to a better outcome. So what's the strategy, the approach that we have been using at QCRC? The first and most important step, patient selection. And this was initiated with a center here in Qatar, extending in the region and even beyond at the intercontinental level. And I will show you the different locations in which we have now set up clinics and recruit patients. The next important step in this approach is how do you go about to identify mutations. And to do this, we're using third-generation sequencing, a cutting-edge technology, the most advanced at the moment for sequencing the human genome. And we will talk about that briefly. And very importantly, build a strong team with clinicians, geneticists, molecular biologists, bioinformaticians, all the different expertise that needs to come together in order to address such a complex problem. So to start with an overview of the different clinical centers, with the key center being here in Qatar and extending in the region, Europe and the US, where we now have clinics and have been recruiting patients. The recruitment criteria, very important to ensure that these are homogeneous across the different centers. So for dilated cardiomyopathy, which is the focus of today's talk, inclusion criteria include LV dilatation, reduced ejection fraction. Exclusion criteria, coronary artery disease, alcohol abuse, or thyroid disease. For hypertrophic cardiomyopathy, again, inclusion in criteria, increased wall thickness, mass, as long as this is not caused by pathological loading conditions. And exclusion criteria, there's a number of those: hypertension, valvular disease, etc. We're also recruiting patients with ARVC using the international criteria for that, which were published in our latest article in the journal that you can find a copy of outside. And for all those patients, in addition to the clinical history, we make sure to have echocardiography results, cardiopulmonary test, stress test, cardiac MRI, and coronary angiography results, so that we can include these in the statistical analysis and compare with all the genetic findings. So at the genetic level, what technology do we use? It's called third-generation sequencing, or you'll find it in the literature as next-generation sequencing as well. What is this? You've probably heard of the term sequencing and sequencing approaches being able to read base by base the entire genome. Well, up until a few years ago, this was a very tiresome and time-consuming procedure. So to decode the entire human genome around 2000, something you've heard on the news. That took 10 years. It took about $3 billion and labs from all over the world to decode the human genome from one person. Today, you can read the entire genome of several people in 10 days with a cost of about $3,000 per genome. So what drove this tremendous change? The development of this technology, third-generation sequencing. So this is what we're employing now to screen the DNA of our patients, because it will be much faster, much more economical, and can yield important results for what we've handpicked to be 201 genes that include all known genes to be involved in heart disease, and a large number of candidate genes that we've selected because of their function in the cardiomyocytes in the heart. So therefore, they're very promising in this context. This adds up to about 1.5 megabases of DNA that we are currently screening for each one of the patients in this study. The platform we used, because there are now several different technologies that do third-generation sequencing, what we chose to use is the Illumina HiSeq machine. Why? Well, there's increasing evidence in the literature with a recent paper in 2012 showing that this technology has not only a lower error rate, appears to be least biased coverage across the entire genome, as well as the repetitive sequences of our genome, and among the next generation sequencing systems, this ranks as a top performer. So using this approach, we went on to follow the method that I'll quickly take you through. So what does third generation sequencing do? We take blood from the patients and extract the genomic DNA. That's broken into small pieces, and all these little pieces are sequenced in parallel. That's what makes it really fast. You sequence all those small parts in parallel. You align the sequences and using bioinformatical approaches, and you end up reading the exact sequence of that individual for each one of those 200 genes. To break this down into pieces, the first step is to take your genomic DNA and cut it down into small pieces. But you need to have a way to identify those pieces so that you can read them. And this is why we label them with specific adapters. You can imagine them as a colored adapters. Not really, but for the purposes of the presentation. So what you end up is every little piece of DNA has an adapter on it, and this is what's called a library. So you make a library of the DNA from each patient. These parts of the library are pulled together, but if you want to screen for specific genes and not everything in there, those 201 genes, then you need to pick out the regions from the entire genome that correspond to those 200 genes. So how do you do this? Well, you create probes with little biotin molecules on them. Those probes can bind specifically to those areas you're interested in, your target genes. And then, using streptavidin beads, you fish out, if you want, all those regions that you're interested in. So by fishing them out, you end up with what's called an enriched library that contains only those regions that you want to sequence. So this makes it even faster because you don't have to screen our 3 billion bases of our genome, but only those 1.6 megabases that we're focusing on. Another thing that makes this really fast is the fact that if you use different colors to label the genome of each patient, you can sequence multiple patients at the same time. So you can have person A being labeled with yellow, for example here, the DNA from patient B with purple, etc. You mix them all together and you can still distinguish them at the end of the day because you have used different colors. So, results today. What have we achieved so far at QCRC? First of all, prepare the research protocols that we are applying to collect patient samples. Get ethics approvals from all the different clinics for all the different countries that are involved. Establish the cardiomyopathy clinics. Ensure that we have informed consent procedures with all the detailed information provided and explained to the patients before they're recruited. Creation of a clinical database, a crucial part of the process, a clinical database where we import all the details for each patient, anonymously of course, with identifiers, so that we can later on perform the statistical analysis, and then the standard operating procedures both for the blood and DNA collection, storage, quality control, etc. So this led to the establishment of the DNA bank that's continuously being enriched. How about patients? How many patients have we recruited today? So we have approximately 450 hypertrophic cardiomyopathy samples, 42 dilated cardiomyopathy, and that includes sporadic and familial cases, 2 ARVC cases, as we only recently started recruiting those. From all those cases, we've already sequenced and bioinformatically analyzed 28 DCM cases, and I'll show you the results in a minute, 16 HCM patients. In the machine right now, being sequenced, another 150 HCM cases, and the new samples that are in progress, 27 HCM, 14 DCM, and 4 family members. It's very important to note here that in addition to as many affected members from each family as possible, we're trying to recruit as well unaffected family members, because they serve as the negative control. If you find something seemingly pathogenic in an affected member, and then you also find it in the unaffected, clearly it has to be ruled out. So, the sequencing results we have so far. Very happy to show you that some of our findings have immediate clinical value. Specifically, in 2 families, we identified mutations in phospholamban and lamin A. These mutations have been reported before in patients with dilated cardiomyopathy. So this is of direct and immediate clinical value because these are known to cause the disease. They're important for the family of those patients to know, because it could enable pre-symptomatic testing if so desired by the family. And these families are now being provided with genetic counseling to explain to them what this is, what the cause of their condition is, and what it means to them and their family members. Results of immediate research value. Over here, I'd like to start by pointing out 2 likely pathogenic variations that we have identified in the gene of titin. So titin is the largest human protein. In actual size, it's about 2 micrometers long, and for many it's considered a giant spring in the heart. It's a structural protein and it's crucial for muscle contraction, as we'll see in a minute. Titin is mutated in about 18 to 25% of DCM cases, so it's known to be implicated in the pathogenesis of this disease. However, what we have identified, those 2 variants, are novel, have never been described before, and importantly, when using bioinformatical tools, these mutations, variants are predicted to actually lead to a pathogenic outcome because they affect the size of the protein. So one of these variants actually leads to a much shorter, about 50% shorter titin molecule, whereas the other leads to a larger molecule because of a duplication in its region. So here is titin. This is your sarcomere, the key contractile structure in cardiomyocytes in the heart. So you have titin, the large molecule extending from the Z line to the M line. So you can appreciate that mutations that change the size of this protein could really affect how the muscle contracts. Previous studies from our team led to a publication in the New England Journal of Medicine in 2012 showing how common and important mutations in this gene are for dilated cardiomyopathy patients, and we actually have one more publication that will be coming out soon with more details on titin in DCM. Some more findings, novel, possibly pathogenic variants in 2 other genes, desmoplakin and desmoglein-2. Why are these important? Again, these genes and their proteins are crucial for the structure of the cardiomyocytes and cardiac function. Why they drew our attention? Because the mutation that we identified in desmoplakin was present in 2 affected sisters from the same family. We're now screening more affected members and unaffected members from the same family. We have the MRI pending, and importantly, the bioinformatical tools are predicting that this mutation leads to an abnormal protein. And the same holds for desmoglein too. So if we were to look diagrammatically where these proteins lie, you have desmoplakin and desmoglein over here in the desmosome structures that are crucial for the communication of adjacent cells. There's many more results I could be talking to you about, but we're going to run out of time very, very soon. So I'll just point out that we have identified more than 50 novel variants, in addition to the ones I just talked about, in over 15 different genes. Representative ones are here. All of these are brand new, have never been described before in the literature, and we will work our way through them gradually to determine what these changes mean. Overall, we identified over 1,600 variants in each of the patients that we screened so far. Thankfully, many of those variants were known, so we don't have to screen all 1,600 of them for every patient. So, what lies ahead? Our plans for the future. Importantly, continue with the clinical aspect of this work, the recruitment of patients from all different continents, with the center being here in Qatar and extending to the different regions of the world, which is crucial so that you can compare different populations and have a better sense of what these mutations do in the context of different ethnical backgrounds. Proceed right away. We have started with in silico modeling in order to predict what these mutations are doing to the protein structure, and from there on, extensive in vivo studies with iPS cells from those patients and in vitro studies both at the cellular and molecular level in order to determine the exact mechanism that leads from the mutation to the disease. And with that, I'd like to thank the many, many members of our team, especially from here in Doha, the clinical and the laboratory teams, the team in Egypt, Athens, Florence, London, US, and our operations team who is making all this possible. And above all, I'd like to thank Professor Sir Magdi Yaqub for joining us all in this dream, this amazing work. That it's really an honor and pleasure to be joining, and Professor Bobono for all his incredible help in making this a reality. Thank you.